Bioinformatics. What is bioinformatics? It's a discipline that allows to apply tools of informatics for understanding of biological data. It combines computer science, biology, mathematics, and engineering. It's a multidisciplinary science. Bioinformatics has become an important part of many areas of biology. In molecular biology, bioinformatics techniques such as image and signal processing allows extraction of useful results from large amounts of raw data. In genetics and genomics, it aids in sequencing and annotating genomes and their observed mutations. At a more integrative level, it helps analyze and catalog the biological pathways that are an important part of system biology. In structural biology, it aids in the simulation and modeling of DNA, RNA, and proteins, as well as biomolecular interactions. Bioinformatics is based on computational programming and sequencing. Computational programming uses advanced computing capabilities to understand and solve complex problems, with algorithms to solve science, engineering, and humanity problems, and computer and information science to optimize the advanced system hardware, software, networking, and data management. Sequencing is to determine the primary structure of an unbranched biopolymer.
Now we will learn a bit about DNA sequencing. DNA sequencing is a very important tool to improve biological systems, but before entering the sequencing, we will make clear some concepts. The DNA is deoxyribonucleic acid and is the hereditary material in humans and almost all other organisms. DNA is a molecule that is made up of a pentose, which can be ribose or deoxyribose, depending on whether it has a group OH or an H on carbon 2, and monophosphate, diphosphate, or triphosphate depending on the number of phosphates it contains. It also has a base, which can be purine, either adenine or guanine, or pyrimidine, whether cytosine, thymine, or uracil. Both are linked by a glycosidic link. How to mark a molecule or a base for sequencing. The DNA is marked with fluorophores. Each sequence is given a fluorophore and given a specific wavelength so that they emit light and can be detected. Principle of the solid sequencing system. From the sample to be sequenced, a library of DNA fragments is prepared by sonication or physical disruption, which will be used to prepare populations of clones in magnetic beds. The joint fragments carry a universal adapter sequence P1, so that the initial sequence of each fragment is known and identical. Then a PCR is performed by emulsion in microreactors, with water in oil containing all the necessary reagents. The PCR products are selected by size to obtain a suitable size and are covalently bound to a crystal. Primers are added that hybridize with the adapter sequence of the library mold. 4 types of fluorescent labelate, 2 base proofs compete for binding to the first used for sequencing. The specificity is achieved by interrogating each first and second base in the ligation. Reaction. Several cycles of ligation, detection, and breakage are performed. After 5 cycles, with 10 ligation reactions each, the extension product is removed and the mold is reused, with a first complementary to the N1 position for a second round of cycles. 5 rounds of cycles are used for each sequence. With these repetitions, each base is interrogated in 2 independent ligation reactions by 2 different primers. Sequencing technologies. Today, thanks to technology, there are many specific devices and kits that help us in the work of sequencing. Its use depends on the objective of the project you want to achieve. How is a genome stored in a computer? First, they have to convert the digital code of ones and zeros to a genetic code of A's, C's, T's, and G's. Then take this lowly text file and manually construct the molecule it represents. Each of these is a fitting and of itself. DNA storage requires cutting-edge techniques in data compression and security to design a sequence both info-dense enough to realize DNA's potential and redundant enough to allow robust error checking to improve the accuracy of information retrieved down the line.
Web tools necessary for gene research, functionality prediction, and primer design. There are several tools on the internet available for free that can be used as search engines and databases for the research of certain nucleotide sequences that have been previously studied. Some of the most commonly used databases are the National Center for Biotechnology Information, the DNA Database of Japan, and the European Bioinformatics Institute. There are also web tools and databases available for proteins, like UniProt and PFEM. For example, UniProt has the objective to provide the scientific community with a comprehensive, high-quality, and freely accessible resource of protein sequence and functional information. It contains a large amount of information about the biological function of proteins derived from the research literature. The NCBI is part of the National Library of Medicine of the United States. It houses a series of databases relevant to biotechnology and biomedicine, becoming an important resource for bioinformatics tools and services. Major databases include GenBank for DNA sequences and PubMed, a bibliographic database of the biomedical literature. The NCBI also has available a useful tool called BLAST, which is used for comparison between nucleotide sequences or proteins. BLAST calls for basic level alignment search tool, and it finds regions of similarity between biological sequences. The program compares a sequence with multiple databases and calculates the statistical significance. BLAST can be used for several purposes. These include identifying species when working with DNA sequence from a known species, locating known domains within a sequence of interest, DNA mapping when working with a known species, and looking to sequence a gene at an unknown location.
We can access to many different databases available in the internet using common web browsers just like Google Chrome, Firefox, and Safari. There are several databases available depending on what we are looking for. For example, for protein structure, we can enter to these two databases named Worldwide Protein Databank and the Protein Databank. In the case of domains and motifs, we have all the information available on UniProt and Interpro. The most used databases for primer design are Integrated DNA Technologies and SnapGene Viewer. To measure the molecular evolution, we can use neighbor joining and UPGMA.
The climate change has been affecting the world for a lot of time, causing a lot of troubles like global warming. This at a time causes drought, and the drought causes the death of the plants. Let's use genetic engineering to solve it. Here we have the solution. First, you use a plant with genes that give resistance to drought. Then, you took these genes and you put them in a new plant. But how we're going to obtain our gene of interest? Well, we're going to use NCBI. Step 1. You have to open NCBI on your computer and look what you're going to search. Now, we are going to use gene and expression for our project. Step 2. You have to put on the searcher what are you looking for. In our case, drought plants. In the left side, you're going to see a lot of options. You have to put genes. Step 3. Now, you only have to choose one of the genes that responds to stress. In this case, the stress is the lack of water. You can also use proteins. You have to go back to step 2 and instead of put genes, you use proteins.
There are several examples of how to apply bioinformatics in genetic engineering, like the investigation done by Dr. Luis Figueroa. Plants respond to stress through metabolic and morphological changes that increase their ability to survive and grow. To this end, several transcription factor families are responsible for transmitting the signals that are required for these changes. In the study, the transcription factor superfamily AP2ERF, particularly the gene RIP2.4 from carica papaya, was isolated 4 genes, and in a silico analysis showed that the 4 genes encode proteins that contain a conserved AP2 domain located within group 1 and 2 transcription factors of the AP2 superfamily. Semi-quantitative PCR experiments indicated that in each CPRIP2 gene is differentially expressed under stress conditions, such as extreme temperatures. Moreover, genetic transformants of tobacco plants overexpressing 2 genes show a high level of tolerance to cold and heat stress compared to non-transformant plants. Confocal microscopy analysis of tobacco transgenics plants show that the gene CP-RAP2.4 proteins were mainly localized to the nuclei of cells from the leaves and roots and also in the seed elements. Moreover, the movement of this RNA in tobacco was analyzed. The results indicate that the gene in tobacco graphene was analyzed in the papaya tree have a functional role in the response to stress conditions such as exposure to extreme temperatures via direct translation outside the parental RNA cell. In recent years, investigators have successfully developed super efficient systems using alternative technologies to generate genetically engineered mice much faster and more economically compared to traditional targeted mutation methods. The CRISPR-Cas system has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate, and more efficient than other existing genome editing methods. CRISPR-Cas9 was adapted from naturally occurring genome editing system in bacteria. The bacteria capture snippets of DNA from embedded viruses and use them to create DNA segments known as the CRISPR arrays. The CRISPR arrays allows the bacteria to remember the viruses. If the viruses attack again, the bacteria produce RNA segments from the CRISPR arrays to the targeted viruses, DNA. The bacteria then use Cas9 or a similar enzyme to cut the DNA apart, which then disables the virus. The CRISPR-Cas9 system works similarly in the lab. Researchers create a small piece of RNA with a short guide sequence that attach to a specific target sequence of DNA in a genome. The RNA also binds to the Cas9 enzyme. As in bacteria, the modified RNA is used to recognize the DNA sequence. Although Cas9 is the enzyme that is used most often, other enzymes can also be used, and the Cas9 enzyme cuts the DNA at the targeted location. Once the DNA is cut, researchers use the cell-sound DNA repair machinery to add or delete pieces of genetic material or to make changes to the DNA by replacing an exciting segment with a customized DNA sequence.